The subject of human genetic enhancement, also known as human genetic engineering, revolves around elevating the human condition through modifications at a genetic level. The objectives for such modifications could range from curing diseases, mirroring the preventive nature of vaccines, enhancing athletic prowess, modifying physical appearances and metabolism, or even amplifying physical and mental capabilities like memory and intelligence. However, the heritability of these genetic enhancements has been a point of contention within scientific circles. The practice of genetic modifications aimed at curing genetic diseases is referred to as gene therapy. There is a growing body of gene therapies that have been developed, tested in clinical trials, and approved by the FDA. Between 1989 and December 2018, over 2,900 clinical trials took place, with more than half of them reaching phase 2 noteworthy therapies to make it to the market are Luxturna by Spark Therapeutics for the treatment of RP65 mutation, induced blindness, and Novartis Kymriah, a chimeric antigen receptor T-cell therapy. Additional drugs from Novartis and Alnylam, among others, have also gained FDA approval both in vivo and ex vivo gene insertions, typically use adeno-associated viruses AAVs and lentiviruses. Asocerna approaches like those from Alnylam and Ionis Pharmaceuticals utilize non-viral delivery systems, employing alternative transport mechanisms to liver cells via GalNAc transporters. The ethical implications of genetic engineering have elicited fervent debates among bioethicists. Coupled with genetic testing and stem cell research, these topics intertwine in a complex ethical discussion. Central to this debate is the distinction between fixing defective genes and enhancing normal ones. Many argue while gene repair is morally acceptable, enhancement introduces potential for discriminatory eugenic practices leading to social injustice. Ethical issues surrounding gene testing often relate to familial obligations to reveal inherited disorders, medical rights, and responsibilities concerning genetic test disclosure, the controversies surrounding genetic discrimination, and the moral justification of using genetic testing to avoid creating severely disabled individuals. Lastly, the moral and ethical considerations surrounding gene therapy and human genetic enhancement involve an evaluation of the medical risks and benefits, obligations to use the procedures to alleviate suffering, reproductive freedom in making genetic choices, and the accountability in promoting positive genetics, including attempts to enhance normal functions. Gene therapy technologies could potentially revolutionize athletics by enhancing physical performance. While no cases of genetic doping have been documented, certain gene therapies might potentially be used for this purpose. Proponents like Kaiser et al. Believe that granting all athletes equal access to such procedures could make the field more competitive. Critics, however, argue that using gene therapies for enhancement rather than treatment compromises ethical principles in both medicine and sport. Genetic intervention could potentially have many other effects, from altering physical characteristics and metabolism, to enhancing memory and intelligence, to increasing resistance to depression or relieving chronic pain. For instance, it might be possible to prevent certain congenital disorders that affect physical appearance and possibly cause discomfort by altering the relevant genes. Moreover, research is ongoing into behavioral genetic intervention, with a focus on genes believed to influence traits such as aggressiveness or altruism. A related field of research is the potential use of gene therapies to treat psychiatric disorders by altering gene expression in the central nervous system, SCENES, to influence neural regeneration and plasticity, and therefore behaviors. Recent studies have explored the possibility of modifying genes such as ALT2 to reduce alcohol consumption, or PALEVIN, the reduction of which has been found to induce depression, like behavior in rodents. Other studies have investigated cognitive enhancements. For instance, transferring the CBP gene has been seen to increase expression of BDNF and improve cognitive deficits in Alzheimer's animal models, countering the impact of amyloid-β, which interferes with normal memory formation. Finally, research is also being directed into the proto-science of genoeconomics, founded on the belief that financial behavior is genetically determined. While correlations have been identified, results remain inconclusive as of 2015.